Okay, hello everybody. This is a podcasted version of the Translational Regulation lecture, which is part of the Medical Genetics module at the University of Bradford. So the learning objectives for this lecture are as follows. The key thing really is that by the end of the lecture, and by the time you've engaged with wider reading, etc., you should be able to critically discuss the mechanisms, or some of the mechanisms that the cell uses to regulate the translation of mRNA. And you should also be able to discuss how errors in this translational machinery can lead to certain types of human disease. I just want to emphasise at this point that this lecture isn't about understanding the actual process of translation. You've covered translation with regards to how ribosomes work, etc. on other modules, and I don't want to go into repetition of that material or even look at that in more detail in this lecture. What I want to consider in this lecture is how the cell regulates the actual act of translation itself. And thus impinges another level of regulation on gene expression. So to start with, I just want to show quite a complex diagram, which provides an explanation as to how the actual initiation of translation is achieved in eukaryotic cells, in human cells. Because it's actually very different to what occurs in prokaryotes. The actual act of translation, as I've just said, is quite similar between prokaryotes and eukaryotes. There's subtle differences in the size of the ribosome, subunits, etc. But the way that the different sites within the ribosome, the E, the P and the A site, function to add amino acids to the green polypeptide chain, etc. The tRNAs recognise and scan the codons. All that's similar to what you've been taught before in previous modules in years 1 and 2. But the initiation of translation in eukaryotes is very different to prokaryotes. And it's this initiation of translation that's often acted upon by the cell as a means of regulating the process itself. So this is why I want to just give you a little bit of background and detail with regards to how this initiation is initially set up. So as you may or may not recall, in bacterial translation, there's a particular sequence found in the mRNA called the Shine-Dalgarno sequence. And it's this Shine-Dalgarno sequence, this run of nucleotides in the messenger RNAs of prokaryotic transcripts that ribosomes recognise and bind to in order to initiate translation, copying the mRNA codons into protein. Now, human genes, eukaryotic genes, don't have these Shine-Dalgarno sequences present in their mRNA sequence. So the question is, how in the absence of the Shine-Dalgarno sequence are ribosomes correctly loaded onto the start of the 5' end of mRNAs prior to translational initiation? Well, in eukaryotic cells, it's actually this 5' cap structure that's key to the initiation of translation of the mRNA. So we've talked about the 5' cap structure in the previous lectures on mRNA processing, how it's important both in nuclear export and protecting the mRNA from exonuclease activity. Once the mRNA is exported to the cytoplasm, the cap is very important then in the process of translational initiation. So what actually happens at the 5' cap of the mRNA following export into the cytoplasm is that cap binding complex, the CBP20 and CBP80 proteins, which are loaded onto the 5' cap during transcription and post-transcriptional modification, are removed and replaced by a different set of proteins at the 5' cap. The main protein you need to remember and be concerned about is this protein EIF4E. This just simply stands for eukaryotic initiation factor 4E. And it's this EIF4E protein that really can be considered the master regulator of translation, insofar that it is by modifying and affecting EIF4E function that the cell is most frequently able to modify how translational efficiency and the level of translation that occurs in the cells, how that proceeds. So let's quickly look at this diagram. You won't be asked questions directly relating to all of the detail present in this diagram, but you do need to have a conceptual understanding of what's going on to initiate translation, because that's required in order to understand how the cell then regulates that process. So let me just explain what is going on here. So prior to the initiation of translation in eukaryotic cells, what you have is the assembly, side by side almost, of two different components that are going to eventually come together to form what is known as a pre-initiation complex on the beginning of an mRNA. The first of these two components is the small 43S subunit of the ribosome itself. And what happens here is that the small subunit of the ribosome associates with a range of eukaryotic initiation factors, and then also the initiator tRNA, the methionine tRNA, in such a way that it's ready then to locate and bind to the 5' end of mRNAs awaiting translation. So that's this side of things, and you can read about this in the core text. But the bit that I'm actually interested in, the bit that I want you to focus on, is this side of the diagram, which involves the mRNA itself. So what we have here is a 5' cap of an mRNA. This is the start codon, so the ATG, or in the mRNA obviously that is in AUG. You should know by now that thymidines are replaced for uracil in RNA. So here's the AUG, the start codon in the mRNA. And what you also tend to get in eukaryotic RNAs very often are these so-called secondary structures that form within the mRNA molecule. Now these secondary structures can have many purposes, but one of the consequences that can arise as due to the presence of these secondary structures is that ribosomes can't really move efficiently along the mRNA. So there's a requirement to remove these secondary structures from mRNA prior to translational initiation. And that's where these eukaryotic initiation factors come in, particularly EIF4G and EIF4A. So what actually goes on this side of the diagram is as soon as the mRNA emerges from the nuclear pore complex, the cap of the mRNA is modified, CBC20 and CBC80 are removed, and are replaced by EIF4E, which now binds to the 5' cap. Once EIF4E is bound, it then interacts with another initiation factor called 4G, and then 4G interacts with another initiation factor called 4A. So you can think of EIF4G as a bridge that links the EIF4E protein and this EIF4A protein. Once these three proteins have been assembled on the 5' end of the RNA, another initiation factor called 4B gets recruited, and the action of this 4B and 4A initiation factor unwinds this secondary structure in the mRNA. So these are RNA helicase proteins, EIF4A and EIF4B. And these RNA helicases unwind that secondary structure that's present in the RNA, which gets the mRNA in a state where it's ready then to interact with the ribosome and ready for the ribosome to start copying the mRNA codon sequence into protein. So what I really want to take on from this first slide is the role played by this EIF4E protein. If this isn't present, you can't recruit 4G and these 4A and 4B proteins. And quite simply what that means is the mRNA can't be actually altered in such a way that its shape is then conducive for the start of a translation to begin. So there's another thing as well that's important to understand about translation in eukaryotes and how it differs from translation in prokaryotic cells, and that is that the actual structure of the mRNA during translation is not linear. So if you look at this figure here, what this is showing is the circularization that occurs during so-called steady-state translation in an mRNA molecule. So you've not got the situation where an mRNA is linear like this, and then a ribosome binds here and moves along it, and then it falls off the end, and then another one comes on, and it moves along it and falls off the end. What actually happens in the cell is the RNA forms a circle structure, and ribosomes kind of bind onto this structure, move around the structure, and when they reach the end of the mRNA, the stop codon, and fall off, they're automatically in close proximity to the beginning of this process, and thus the ribosome subunits are recycled in an extremely efficient manner. Now this circularized structure requires the action of a couple of proteins which are present on the mRNA. The first protein that it requires is EIF4G, and the second protein it requires is polymerase. Sorry, not polymerase, polyA binding protein. What happens is EIF4G interacts with polyA binding protein, and via this interaction you get circularization of the mRNA. Now, this is a really efficient way of doing translation, but it also offers a cell a unique opportunity with regards to regulating steady-state translation, because you can switch off this circularization of the RNA structure by one of two methods. The first way of switching it off is to interfere with the cap in such a way that EIF4G is not recruited and cannot form this interaction. The second way of affecting translational efficiency is to reduce the length of the poly A tail. If the poly A tail becomes too short, there's simply not enough poly A binding protein present to interact with the IF4G, and again this circularization structure can't form. So the cell can interfere with translation efficiency by either reducing the length of the poly A tail or modifying the proteins present in this 5' cap structure. Indeed, if you look at mRNA half-life, this is often linked in actually with a reduction in the length of the poly A tail. So as mRNAs are hanging around in the cytoplasm, often you can observe a reduction in the length of the poly A tail, and when the poly A tail reaches a certain size, in other words when it's reduced by a significant amount, the efficiency of translation of that RNA is drastically reduced, and then ultimately the RNA is degraded. So let's consider in more detail then how the cell actually goes about regulating this translational efficiency, starting with how it can regulate translational efficiency by modifying this master regulator of translation, EIF4E. So the most common examples actually of translational regulation involve the EIF4E protein. So just to recap, this is the protein which is placed on the 5' cap as soon as the mRNA is exported into the cytoplasm. And remember that EIF4A is required for the recruitment of EIF4G and the subsequent recruitment therefore of these helicase enzymes that unwind secondary structure. EIF4E is also required for circularization of the mRNA during steady state translation, because if you haven't got any EIF4E, then you can't recruit EIF4G, which is a protein that interacts with the poly A binding proteins and to allow that circularization to occur. So the best example of how the cell modifies translation levels is the example provided by the MTOR signaling network. So mTOR signaling tends to occur in response to stimulatory factors such as growth factors, etc. So in other words, if cells are sent signals to divide rapidly, and those signals generally take the form of growth factors which will bind to the cell surface, one of the actual proteins which become activated inside the cell in response to those growth factors binding to the cell surface is a protein called mTOR. And then what mTOR does is it effectively operates within a signaling cascade. So one of these MAP kinase signaling cascades that hopefully you've encountered in other modules. But if not, you can think of them like a domino effect, whereby one kinase protein signals to another kinase protein that signals to another kinase protein, and you can have a domino effect passing through the cell, which ultimately results in an effect being results in a downstream effect, normally a change in some kind of gene expression control. So the mTOR signaling cascade, what this actually does is it ultimately results in the phosphorylation or a change to the phosphorylation state of this protein 4EBP. So 4EBP or 4E binding protein is a very important protein in translational control. So in the absence of mTOR signaling, so in other words, when a cell isn't getting these growth stimulatory signals, 4E binding protein is hypophosphorylated. In other words, it lacks phosphorylation, or it's phosphorylated only to a very small degree. And in this hypophosphorylated state, 4E binding protein binds to EIF4E and interferes with its role in the recruitment of EIF4G. So remember, if EIF4G isn't recruited, you can't circularize the mRNA, and also you can't recruit the RNA helicases that are needed to iron out these secondary structures and allow the ribosomes to latch onto the RNA. So in other words, by preventing the interaction of EIF4E with the EIF4G, you prevent the initiation of translation, and you also severely impact on the efficiency of translation. So when mTOR is activated by growth factors, etc., what happens then is that 4E binding protein becomes hyperphosphorylated. It's phosphorylated at many sites throughout the protein. And in this hyperphosphorylated state, 4E binding protein can then no longer interact with the EIF4E. So this frees EIF4E up to interact with the other proteins it needs to initiate translation. Now there are other aspects to mTOR signaling that don't relate to EIF4E, but as you can see from the diagram, they all directly relate to increased translation and cell growth. So mTOR is indirectly responsible for increasing the amount of ribosome biogenesis and tRNA production. So obviously, if you want to embark as a cell on a period of rapid growth, you're going to need a lot of protein translation going on in order to facilitate that growth, which means you're going to need a lot of ribosomes present, and you're going to need a lot of tRNA molecules present to carry out that protein production. So mTORs kind of oversees the regulation of a whole range of cellular processes, all of which feed into increased translational efficiency and cell growth. But the key example regards to the control of translation itself is this 4E binding protein and how it interacts with and affects function of EIF4E. Okay, so there's other ways in which a cell can also affect translational efficiency that don't rely on it impinging on these eukaryotic initiation factors. Another way is via actually taking advantage of secondary structures that are present in the RNA. So we've already talked about the secondary structures that are present in the 5' untranslated region of RNA that need to be removed for translational initiation to begin. But let's have a think about how these structures arise in the first place. Well, they tend to arise in RNA due to the presence of these so-called palindromic sequences. For those of you who don't know what a palindrome is, or a palindromic sequence is, it's effectively a mirror image that appears in the sequence of the DNA. So here we've got GAA, CGTCC, and then there's a few bases in between, but then we hit the palindromic mirror image of this sequence here, GGA, CGTTC. So if you imagine what can happen here with regards to the formation of secondary structures, if you form a loop out of this linear DNA here, you allow the alignment of these two palindromic sequences. So in other words, this C and this G align, this C and this G align, this T and this A align, and they form base pair interactions in the same way that they're performing base pair interactions here. So this causes the presence then of a stem loop structure, this non-specific single-stranded RNA here, and then you've got the double-stranded RNA present here in the mRNA molecules, the stem loop. So these stem loop structures, these regions of double-stranded RNA, can be used by the cell in order to modify translation efficiency. So a simple example would be the presence of a stem loop in the 5' untranslated region of an mRNA that normally doesn't have an effect on protein production, but if this stem loop structure is bound by a specific repressor protein, then this switches off translational efficiency for the simple reason that ribosome initiation and ribosome binding can't occur. So there's a real world example of this that's very elegant and gives a good overview of how the cell can utilize these stem loop structures to regulate expression at the translational level when required. And this involves ion regulation actually. So this works by virtue of the presence of two different stem loop structures. One stem loop structure in the 5' untranslated region of the RNA, and another stem loop structure in the 3' region in the 3' untranslated region of the RNA. And it works via concerted effort with this protein cytosolic aconitase. So the presence of a the presence of an RNA stem loop in the 5' untranslated region is generally a negative regulatory factor for translation. In other words, if you have a stem loop structure, as we've got depicted here in the 5' untranslated region, and a protein binds this, it tends to exert a negative effect on translational efficiency for the simple reason that you can't get translational initiation occurring. Conversely, when you get stem loop structures in the 3' untranslated region of an RNA, these tend to actually increase translational efficiency. As I mentioned earlier, one of the ways in which the cells actually confer half-life to mRNA and facilitate mRNA degradation is by the 3' degradation of the 3' end of the RNA. And often the presence of these 3' untranslated stem loop structures stabilizes the RNA and therefore increases its translational efficiency. And that's what goes on here in this example. So what we have are mRNAs that are responsible for producing two different proteins. The RNA with the 5' stem loop in the 5' untranslated region produces a protein called ferritin, and the RNA with the stem loop in the 3' untranslated region produces a protein called transferrin. Now, how do these two proteins function in ion regulation? Well, in situations where there is excess iron present in the cell, which can be a very toxic situation for the cell to be in, ferritin is required in order to sequester that iron and make sure it doesn't do any damage to the cell. Yeah. So in situations of excess iron, the cell needs a lot of ferritin around to sequester that iron. Yeah. Conversely, if there's not enough iron around, which is equally a problem for the cell needs to release its stores of this metal in order to maintain the correct equilibrium that's required for cellular functions to proceed normally. And this release of iron is mediated via the action of this transferrin receptor. So in a situation of iron starvation, what you actually want in the lack of iron is you don't want any ferritin around, because you don't want to sequester what is there away from the cell, because the cell needs all it can get. And you actually want lots of the transferrin receptor made, because you need to make sure that any stores of iron that the cell has are made available to the cell if it needs them. So what happens in the case of iron starvation then is that the cytosolic aconitase protein binds to the stem loop regions present in ferritin and transferrin RNA. And in the case of ferritin, because this stem loop is in the 5' untranslated region, the binding of cytosolic aconitase downregulates translation and makes sure that no ferritin is actually produced. In contrast, the binding of this protein to the 3' untranslated region stem loop stabilizes a transferrin receptor mRNA and ensures that lots of transferrin protein is made so that iron can be freed up. Now if we look at the opposite situation where we have excess iron, in this situation excess iron tends to bind to the cytosolic aconitase protein. And once bound by iron, cytosolic aconitase can then no longer interact with these stem loops. So what happens then is, in the absence of cytosolic aconitase binding to this 5' untranslated region stem loop structure, ferritin mRNA is translated at a high level of efficiency. Lots of ferritin is made. And this ferritin then can go about the business of sequestering this dangerously high amount of iron that's present in the cell. Again, in contrast, the release of cytosolic aconitase from the 3' untranslated region stem loop destabilizes a transferrin receptor mRNA and makes sure that no transferrin receptor protein is made. So this switches off the export of iron from the stores that the cell has tucked away for a rainy day. Because obviously in the situation of excess iron, the last thing you want is for the cell to start releasing more from its stores. So it's a really elegant system for managing managing iron equilibrium inside the cell, and it all works via the binding of protein, a single protein, to two differently located stem loop structures in the RNA. So another way in which the cell can regulate translation of mRNAs is via transporting RNAs around the cell. So it's not the case that all mRNAs are instantly translated to protein as soon as they're exported from the nucleus. Some RNAs are actually physically taken to different locations within the cell before then they are translated into protein. One example is a myelin basic protein, which is transported to processes in oligodendrites. So in other words, the RNA is taken to the point in the cell where the protein is actually most needed. So this is a little video that hopefully will play during the podcast showing this process in action. So if you look at the tiny white little spots, what they are fluorescently labelled mRNAs that have been tracked in real time in a live cell. And as you can see, the mRNAs are effectively trafficking along the actin cytoskeleton within the cytoplasm to just one end, the apical end of the cell. So this isn't an uncommon process. I mean, this happens in a lot of different cell types. Classic examples of this are found in Drosophila during actual development of the Drosophila larvae, where there are several situations that require cellular polarity to be established during development. In Drosophila, the proteins that are responsible for actually trafficking these specific mRNAs around the cell include the protein egalitarian, which has a human homologue which is called ELAV. And what this egalitarian protein does is it binds to the mRNA targeted mRNA, and it then also interacts with motor proteins such as dynactin, which then walk along the actin cytoskeleton, dragging the mRNA to the correct location in the cell. The reason it does this, as I've just said, is to grant polarity during development, but there are other reasons for trafficking RNAs around the cell as well. Another good example that's a real hot topic with regards to research at the moment is the function of this FMRP protein, which hopefully most of you now will realise is the Fragile X mental retardation protein. So this is the protein product of the Fragile X gene. So it turns out that the FMRP protein is also involved in mRNA transport, but this time in neurons. So this rather complex diagram I've included here, you don't need to worry actually about this actual aspect of the diagram. What I want you to focus on is this, what's going on here. So this is an axon. In fact, this is a synapse at the end of a neuron. And what's happening is, following transcription of the of different RNAs, specific RNAs, the Fragile X mental retardation protein latches onto these RNAs in the cytoplasm and physically drags them to synaptic junctions in the neurons. It does this via an interaction with the 3' untranslated region of target RNAs. And we're starting to get a larger and larger list now of RNAs which FMRP is responsible for dragging around neurons. But this is a work in progress, obviously. We still don't fully understand, one, what the full consequences of this are, and two, what all the target RNAs in the neurons might be. So what is this doing? What is FRMP doing this for? Well, it's thought to be very important in the process the trafficking of these RNAs by FMRP to different locations within the neuron, particularly to synapse points within neurons, is thought to facilitate a property of neurons that's termed synaptic plasticity. So synaptic plasticity is something that gets neurobiologists very excited. It's basically a term that explains how memories can be established between neural networks in the brain. It's also involves synaptic plasticity in the transmission of pain signals, and how we interpret different types of pain. So it's a property of neurons to effectively change their characteristics at a synaptic junction, and by changing the characteristics, this is one way in which we think memories are established and maintained. You can have short-term changes, which might last for a few seconds, and you can have long-term or even permanent changes to what's going on at the synaptic junction of neurons based on this synaptic plasticity. And it's currently thought that FMRP might be very important in establishing this during the formation of neural networks in the brain. Very exciting area. So another real key area then of translational regulation revolves around RNA interference. So you guys had a lecture on regulatory RNA from me in molecular genetics last year, during which we mention microRNAs and short interfering RNAs and RNA interference. But I just want to go into more detail in this lecture about how those processes work. So we've probably actually been observing RNA interference for many years, possibly for many decades. But it wasn't until these two guys, Fire and Bellow from Cold Spring Harbor in New York, it wasn't until these two published some work about 10 years ago on RNA interference that really the full extent of how prevalent this mechanism is within cells and the actual impact it could have on modern biology became really fully appreciated. So what RNA interference is in a nutshell is the ability to modify translational efficiency by acting on the actual mRNA sequences themselves. And this works via the action of two different types of small non-coding RNA, these so-called short interfering RNAs, and then these so-called microRNAs. So let me just recap on what a non-coding RNA is, just for those of you who can't remember what we talked about in April in molecular genetics. In addition to mRNAs, which are termed coding RNAs because they code for proteins, the cell also transcribes, RNA polymerase II also transcribes a whole host of RNAs that actually don't code for anything, that don't make any protein. And we've known about these RNAs for many years, but for over those years, the majority of scientists kind of believe that these non-coding RNAs weren't really important, that there were probably non-specific transcripts that RNA polymerase II just happened to copy by mistake, and then eventually these will be degraded and gotten rid of. It seems foolish now to think that, to dismiss it in that way, but that's what people did for many years. But then gradually we started to get more understanding about how these short and long non-coding RNAs might be impacting on biology. I'm not going to mention long non-coding RNAs today. We mentioned that in molecular genetics. Today I'm going to focus exclusively on short interfering RNAs and microRNAs. If you want to read about long non-coding RNAs, then you can obviously do that in the core textbooks and look for journal articles on those particular molecules. But the lecture is going to focus on short interfering RNAs and microRNAs today. So short interfering RNAs can be derived either from double-stranded RNA naturally, or you can actually produce these artificially in the lab and use them in experiments. MicroRNAs are derived from genomic sequence, and they actually arise from a precursor called pre-microRNAs. We'll go into that in more detail in a second. Both of these function in a very similar way, and what they do is they target mRNAs specifically and affect that mRNA's translation via the action of a multiprotein complex called the RNA-induced silencing complex, or RISC. And this process is summarised in this nice figure here from the core textbook, Molecular Biology of the Gene. So what we have here is we have a double-stranded RNA molecule that finds its way inside the cell. And we also have here one of these precursor microRNAs structures which originate from the genomic sequence of the cell. In both instances, whether you're talking about a short interfering RNA or a microRNA, both of these short non-coding RNAs are initially processed by the same enzyme dicer. So this enzyme dicer takes a double-stranded RNA, or it takes a pre-microRNA, and it chops it up in such a way that you generate a double-stranded piece of RNA that's around 22 nucleotides long. So the size is very specific. It's not by chance that these things end up 22 nucleotides long. The enzymes that do the cleavage cleave in such a way that the microRNAs and siRNAs are always 22, sometimes 21, but always between 21 and 22 nucleotides in length. Once you have cleavage of the microRNAs or double-stranded RNA to produce these short non-coding RNAs, these structures are then associated with this RISC complex. This RISC complex then becomes active via the degradation and ejection of one of the two strands of this microRNA or siRNA. So what you're left with then is you're left with this guide strand, so-called guide strand. So the RISC complex effectively contains a single-stranded short piece of RNA that will have sequence complementary to a specific target mRNA. So what the RISC complex does then is it seeks out and interacts with the target mRNA by virtue of the complementary sequence that's present in the microRNA or siRNA here. Now, there are two possibilities with regards to what happens to that mRNA target, depending on the level of sequence complementarity that's present when the RISC complex binds to the target mRNA. And by that I mean that if all 22 nucleotides present in the microRNA or siRNA match identically with the target RNA, so in other words, if there's complete complementarity, what tends to occur there is that the target mRNA is cleaved via enzymic activity within the RISC complex and then degraded and chewed up. Yeah, so it effectively stops translation dead, and irreversibly it gets rid of the mRNA. There's an alternative situation that can arise where rather than being degraded, the RISC complex leads to the inhibition of translation of the target mRNA. And in these instances, it's thought that the mRNAs are trafficked to so-called P bodies within the cytoplasm, processing bodies, where they're kind of stored in stasis, prevented from being translated until such a time that occasionally they can be released and then translated again. So RNA interference, it can lead to the degradation of mRNA targets and obviously then the downregulation of translation, or it can lead to the repression of translation. If the match is perfect between a microRNA and siRNA and its target mRNA, the target mRNA is chewed up and degraded. If the match is less than perfect, say 18 of the 22 base pairs in the RISC complex match with the target mRNA, then that target mRNA tends to be repressed rather than degraded. There's another way in which RISC can affect gene expression, and that's by being directed to actually back into the nucleus and directed to regions of the chromosome where it can affect chromatin remodeling in such a way that genes are switched off by virtue of methylation to promoter regions or changes to the actual genome architecture, to the way that the accessibility to that particular gene. There's a very clear examples of this happening in lower eukaryotes such as yeast, where we know that RNA interference is important in silencing particular regions of the chromosome. There's less evidence of this at the minute in mammalian cells, but one or two examples are starting to emerge in the literature. Also, the efficiency of this process, the efficiency of RNA interference is staggering. You actually need very, very little short interfering RNA or microRNA to have a very big effect on translation. So you need very few copies initially of these molecules in order to really hammer and shut off translation. And one of the reasons that it works so efficiently, people think, is because there's actually a process built in the cell that allows the amplification of these sequences once the RISC complex is bound to the target. Again, there's good examples in yeast of this amplification process occurring. Less examples at the minute in eukaryotic cells, just for the simple reason that it's far more difficult to do molecular biology in mammalian cells than it is in yeast cells, which is a simple model organism. So we're playing catch up at the moment with regards to mammalian biology and how these different aspects of RNA interference are working, but we are getting there slowly. It's just taking longer because it's a more difficult system to work with. So it's a complex process, RNA interference. There's a really good chapter in molecular biology of the gene on this, and I would urge you to read that if what I've just told you isn't quite clear. So what I want to consider now then are microRNAs, which from a disease point of view are more important, one would argue, than short interfering RNAs. So microRNAs are interesting. They display a particular type of secondary structure, this so-called hairpin loop structure, but with a few differences to what we've talked about thus far with regards to these hairpin loops that appear in the 5' and 3' untranslated regions of mRNA. So what we're looking at here are different microRNAs. These are microRNAs from C. elegans, which was the first organism to actually have microRNAs isolated and identified and these are human microRNAs. These have actually been identified via bioinformatics. And the reason you can do that, the reason you can identify potential microRNAs by just looking at sequence, is that we now have software that can scan the sequence of an mRNA molecule and predict where secondary structures might arise by identifying, in effect, palindromic regions of that sequence. So these have been predicted to exist by bioinformatic techniques. You can confirm whether these exist, obviously, by doing reverse transcriptase PCR and looking for the expression of these actual RNA molecules in a cell, which is what people have to do to confirm that their predicted microRNAs actually exist in real life, inverted commas. So this is what they look like. They've got this hairpin loop structure. You can find microRNAs effectively anywhere in an mRNA transcript or an RNA transcript. So any transcript that's produced by RNA polymerase II may contain microRNAs in it. And by anywhere, I mean that these stem loop structures can occur in a coding region of a gene. They can occur in the non-coding region of a transcript. They can occur in the intronic sequence. They can occur in the exonic sequence. So they can occur anywhere within a piece of RNA that RNA polymerase II has produced. So this is another reason why intronic sequence is no longer considered junk. It's not just because of its involvement in alternative splicing, like we talked about in the previous lecture. It's also because a lot of the intronic sequence of our genes contain these hidden, if you like, microRNA sequences, which we're now starting to appreciate are very important with regards to fine-tuning translational regulation. So how do we get from these stem loop structures in RNA polymerase II transcripts to the 22 nucleotide microRNA that is loaded into that RISC complex and then targeted to specific mRNAs for degradation, etc.? Well, there's two processing events that are required. In this state, so when the stem loop is present as part of a longer RNA transcript, this is called a pre microRNA. And you require a processing event to occur to convert this into a pre-microRNA, which effectively severs this stem loop from the RNA transcript and frees it up. Once you've severed the stem loop and freed it from the rest of the transcript, there's then a second processing event that occurs that converts this stem loop structure into this 22 nucleotide double-stranded microRNA structure. So the first step, this step here, is performed by an enzyme called DROSHA that's unique to microRNA processing. DROSHA isn't involved in short interfering RNA processing. Once DROSHA has produced this first cleavage event to free the stem loop from the full transcript, excuse me, a second event occurs by virtue of this enzyme called DICER. And what DICER does is it chops up this stem loop structure to produce a 22 nucleotide microRNA. So this is shown nicely here on this diagram of a microRNA. So the microRNA is composed of several segments. There's a single-stranded piece at the head, if you like, or the cap of the microRNA stem loop, which isn't paired up. And then there's an upper stem region, which is where the vast majority of palindromic sequence is. This is actually the part of the pre-microRNA that will eventually be inserted into the RISC complex and used to target mRNAs. And then there's a lower stem loop structure and basal segment to that microRNA. So what DROSHA does is DROSHA cleaves the actual microRNA in this lower stem region here, severing it from the longer transcript. So remember, a long transcript will run like this, and then you'll have a stem loop coming off of that, and then the long transcript will continue. DROSHA comes along and it cleaves this stem loop structure off of that long transcript. Once DROSHA is cleaved here, the pre-microRNA is then exported into the cytoplasm, where DICER comes along and cleaves its head off. Sounds a bit brutal, but that's what happens. DICER cleavage occurs by virtue of recognition of this terminal loop. So DICER recognises this terminal loop region on the pre-microRNA, binds to it, and then severs the pre-microRNA in a very specific way to leave just the upper stem region, the 22 base pairs that are required to form the mature microRNA sequence. Once you've got this 22 nucleotide mature microRNA, that's then loaded into this RISC complex. So how does that work? Well, this works by virtue of a specific protein family called PAS. So these PAS proteins and PAS domains within these proteins recognise a 3' end of RNA. And in this instance, they specifically recognise the 3' ends of these 22 nucleotide microRNAs. So the PAS domain binds to the microRNA. One of the two strands of the microRNA is then ejected from the RISC complex, leaving just this guide strand. And then this entire RISC complex seeks out the target mRNA molecule, which is then incorporated into the RISC complex by virtue of complementary base pairing across this region of the microRNA, this region here. So whatever the sequence of your microRNA is here, there'll be a complementary sequence somewhere in the cell that's present in an mRNA molecule. That mRNA molecule will be found, it will interact with this region of the microRNA, and if that interaction is perfect, cleavage will occur of the mRNA molecule, which obviously then degrades the mRNA and stops it being translated into protein. So again, you can get a lot more detail if you want in the core textbooks about this process, but in a nutshell, that's how RNA interference works. So to finish with them for the last 5 minutes or so, I'd just like to talk about another way in which the cell is able to regulate efficiency of translation, and that's actually by monitoring and influencing the actual degradation of mRNA itself. So this is less of a targeted mechanism for degrading mRNA like we see in the RNA interference example. This is more generic degradation of RNA molecules. So in that respect, it links to general mRNA turnover. But there is a more directed system in place which can spot errors in mRNA, and then if an error is detected, prevent the translation of that error-prone mRNA into protein, with the hope of obviously not producing proteins of aberrant function. So there are 3 main ways in which general mRNA turnover occurs. There's a so-called decapping pathway, the deadenylation-dependent pathway, and an endonucleolytic pathway. So we've talked about the importance of the cap and the tail on preventing degradation, and they do prevent the action of a whole range of just non-specific exonucleases that are flying around inside the cell. However, occasionally the cell decides it wishes to actively get rid of mRNA, and to do that it needs to get rid of the cap or get rid of the tail. So it can do that by virtue of these so-called decapping enzymes. So what these guys do is they come along and they get rid of the cap of the mRNA. Once the cap of the mRNA is gone, this mRNA then is at the mercy of these exonucleolytic pathways, these exonucleases, which will come along and very rapidly degrade the RNA molecule. Similarly, if the poly-A tail of an mRNA becomes too short, it becomes prone then to the action of these 3' to 5' exonucleases. So these are, if you like, enzymes which will degrade RNA in a 3' to 5' manner. But normally they can't do that because the poly-A tail and its associated proteins protects the RNA from that particular enzyme. If the poly-A tail becomes too short for one reason or another, then these enzymes can get access to the RNA and can degrade it in a 3' to 5' mechanism. The endonucleolytic pathways tend to just obviously cleave the RNA away from the cap and away from the tail. Once you've got a break in an mRNA molecule like this, then a big exonuclease called the exosome comes along and just chews up the cleaved mRNA molecule. So it's the exosome that would also get rid of RNAs that have been cleaved in this manner by the RISC complex as part of the RNAi process. So all three of these pathways are involved in general mRNA turnover. So what about this directed mechanism then? So there's lots and lots of mechanisms present in the cell that monitor sequence fidelity and make sure that DNA replication occurs properly, that makes sure that transcription occurs accurately. There are also mechanisms that ensure that once the mRNA has been transcribed, exported, and it's been translated by the ribosome, that there aren't errors that shouldn't be there, particularly errors that result in premature stop codons or the absence of stop codons. So if we consider what goes on during translation of a normal mRNA molecule, what a newly exported mRNA molecule actually looks like is this. So this is a 5' cap, this is a poly-A tail. I don't know if you can see on this, but there are little joins here between this coding region. These joins represent exon-exon boundaries. So this is where the two exons have been spliced together. Wherever two exons are joined together, you always find a group of proteins associated at that exon-exon boundary called the exon junction complex. This is a group of proteins that's left behind as a deposit by the spliceosome following joining of these two exons together. And for many years people thought that the exon junction complex was important in nuclear export. We now know that isn't the case, but it does seem to be important in regulation of translation. So how does it work? Well, it works by the virtue of, let's first consider what's going on here. So these exon junction complexes are present on a newly exported mRNA. As that newly exported mRNA is translated by the ribosome, these exon junction complexes are kicked off of the mRNA. And eventually the ribosome will move along the mRNA, entire length of the mRNA, removing all of the exon junction complexes, and then it will encounter the termination codon, the stop codon here, which is a signal for the ribosome to stop making protein and to dissociate and release its polypeptide chain. So what happens, therefore, in a situation where, due to mutation that for some reason has not been detected previously, there's a nonsense mutation event has been inserted into the mRNA. In other words, there's been a mutation to the sequence in such a way that a premature stop codon has been inserted into the mRNA molecule that would or should result in the production of a truncated version of that protein. Well, in these instances, there's a mechanism called nonsense-mediated decay that can monitor and prevent the expression of these truncated proteins. And it works thus. If you think about it, the ribosome knows, as far as the ribosome can know anything, the ribosome knows that it should never encounter a stop codon upstream of an exon junction complex. That is, if an exon junction complex is appearing every exon-exon boundary, when you get to the stop codon, which should be at the end of the last exon, there should never be another exon junction complex further along than that. Yeah, because the stop codons are present in the last exon in the mRNA. Obviously, in the case of a premature stop codon, you will have exon-exon junction complexes, or you're likely to have exon-exon junction complexes further downstream than this mutation. So what happens in nonsense mediated decay is that if the ribosome encounters a stop codon telling it to stop translation, the exon junction complex that's immediately downstream of this mutation sends a signal to the ribosome, and the ribosome then causes the decapping of the mRNA that it's currently translating and the degradation of the mRNA by a 5' to 3' endonuclease. So the proteins that are involved in this process is called the UPF proteins. These are associated with the ribosome and the exon junction complexes. And it's a really elegant way of making sure that any mRNAs that contain a premature stop codon are not translated. So what's about this non-stop decay system then? So another possibility is that you have an mRNA produced that doesn't have a stop codon for some reason. Now this is equally problematic because a ribosome doesn't know when to stop making protein. It may end up translating sequence in the 3' untranslated region of a gene, which results in a protein that's got an aberrant function again that could cause disease. So in these instances, the cell relies on two things really. It relies on the stalling of the ribosome at the end of the mRNA molecule. So as soon as the ribosome hits the poly A tail, which it should never get to because it should always encounter a stop codon before that point, but if that stop codon is absent, eventually the ribosome will get to the poly A tail. And when it reaches a poly A tail, the ribosome will start to stall. It'll slow down. Now this stalling event, this stalling of the ribosome, sends a signal via a protein called SCI7, telling the ribosome that this mRNA actually contains a mutation, it's wrong and it shouldn't be translated. So at that point, once this SCI7 protein sends that signal, the ribosome dissociates from the mRNA, and a special exonuclease called the exosome comes along and chews up the mRNA that the ribosome has just dissociated from. Now the system doesn't just stop there. This SCI7 protein also triggers the action of a specific protease that will detect the presence of polylysine at the end of a polypeptide. So the codon AAA, if you get 3 A's in a row, will encode for a lysine residue. If the cell detects a protein that has many lysine residues at the end of the polypeptide chain, in other words, a protein that's been produced by a ribosome which has been translating a poly A tail, this specific exonuclease will come along and degrade that particular protein. So the non-stop mediated decay system doesn't just degrade the RNA that's wrong, it also degrades the protein that the ribosome has made. Because it doesn't want a protein with a long polylysine tail flying around the cell, because that protein might have a gain of function property that could cause disease. So how do errors in these kind of mechanisms lead to disease themselves then? Well, the most common errors actually relate to errors in the mRNA themselves. So we've not discussed IRESs thus far, but effectively IRESs circumvent the need for a cap, circumvent the need to have a cap and EIF4E present on an mRNA molecule in order to recruit a ribosome. So IRES stands for internal ribosome entry site, and it's a very specialised secondary structure of RNA that can itself recruit ribosomes to initiate translation. So several genes utilise IRESs instead of utilising a 5' cap. We're not sure why they choose to do that, but they do. Viruses also choose to use IRESs in some instances. And a very important gene called MYC, which is a proto-oncogene, opts to use an IRES in order to translate its mRNA into protein. Now occasionally you can detect mutations in this IRES sequence. C to T transversion mutation is particularly common in patients who suffer from myeloma. So a change to this IRES structure results in cancer due to a change in the affinity that the MYC mRNA has for a translational enhancer protein called HNRNPK. So when this IRES is mutated, it becomes very it has a greatly increased avidity for this translational enhancer, so that drastically increases the expression of the MYC mRNA and the production of the MYC protein. Now obviously if you start drastically overexpressing proto-oncogenes and proto-oncoproteins, you go on to develop an oncogene and ultimately cancer. There are examples of mutations in specific subunits involved in translation that lead to disease. So for example, there's a rare disease called vanishing white matter that's a neurodegenerative disorder, and this results from a mutation in the eukaryotic initiation factor 2b. The 2B factors involved in the assembly of the small subunit of the ribosome. It's actually involved. It's a guanine exchange factor, which means it exchanges GTP during the assembly of that small ribosomal subunit. Again, if you want to read about that in the wider literature, that's fine, but I don't require you to fully understand how the small ribosome subunit is assembled prior to translation. I've already explained that. But it's important you understand that a mutation in AF2B, which is a component of that small ribosomal subunit, is linked to neurodegenerative diseases. There's rare examples where you can get mutations in the actual genes that encode ribosomal subunits. And then there's an interesting example actually of a mutation that can occur in the mitochondrial genome, which affects the actual tRNA molecules that the mitochondria produce in order to translate its proteins. There's a particularly nasty disorder that's associated with mitochondrial mutation called MELAS, and it's got a horrendous name I can never remember. I think it's, let me think, it's mitochondrial encephalomyopathy, lactidosis, and stroke-related syndrome or something. Basically, it's spontaneous stroke, and it is a real common event in individuals who possess this mutation. And the mutation relates to a mutation in the actual tRNA for leucine that's present in the mitochondrial genome. But I guess a major player, the one that I really would like to emphasise with regards to human disease, is a role played by microRNAs in human disease. And we're now aware that both increases and decreases in microRNA levels can directly result in human disease, in particular cancer. So there are examples where the downregulation of microRNAs, such as microRNA 15 and 16, can directly result in an increased incidence of cancer. In this instance, it's chronic lymphocytic leukemia, CLL. And also examples where increases in the levels of a microRNA can result in cancer, such as small cell lung carcinoma. And the current thinking is that these particular microRNAs, so for example microRNA 15, are probably acting as repressors for oncogenes. So in other words, if you get rid of microRNA 15, you lose that control over the oncogene, and this oncogene can then become overexpressed and lead to cancer. So we now know that microRNA 15A, for example, and 16 are directly involved in the repression of two proto-oncogenes, RAS and BCL2. Conversely, there are a group of microRNAs that are also involved in the translational repression of tumour suppressor proteins, so P10, RB2, etc. Normally, these microRNAs ensure that you don't produce too much of these tumour suppressors, but if the microRNA itself becomes overexpressed, such as microRNA 17, 92, what that can lead to is the over-repression of tumour suppressor proteins, which then in turn can open the cell up to the formation of tumours and cancer. The final example I just want to mention again is this interesting FRMP protein, the Fragile X protein, which also, in addition to its trafficking function, has been shown recently to be associated with the RISC complex, suggesting that the FRMP protein might also have links to the RNAi machinery, and whether there is any link between this function and Fragile X syndrome is currently under debate. So just to finish then, the take-home messages with regards to translation and degradation are: translation offers the cell the opportunity to fine-tune protein production. So there's lots of mechanisms that we've seen over the last hour which a cell can use to directly affect how much translation occurs in the cytoplasm. Degradation of the mRNA is involved in a more broad control of gene expression, and also the ability to survey mRNA sequence and ensure that the ribosomes aren't producing proteins from mRNA that contain obvious mutations such as nonsense mutations and premature stop codons, etc. So I hope that proves useful during revision, and as always, watch out for more lectures in this module on my YouTube channel, Boyne at Bradford. Thank you.